Hello biology students, and welcome to video 3 of the mitosis unit. So in this video we're going to be exploring the question, what controls cell division? So in other words, what allows cells to divide and what kind of things stop cells from dividing? So to start off this discussion, we know that most animal cells only divide when they're stimulated. So they only divide when something else, usually a signaling protein, tells them to divide. However, there are other stimuli and other signals that can other than proteins that can also control cell division. So we're going to look through those first. So for example, if I take cells in a lab, so when I say a lab culture, that means a group of cells that I'm growing in a lab, I notice that they only divide if they're attached to a surface. So what this means is that we say that cells are anchorage dependent. They are dependent on being anchored to something if they're going to divide. So for example, if you have a cell floating around in the bloodstream for whatever reason, because it's not anchored to a surface, it's probably not going to divide. At least most cells would not divide in that situation. Another factor that controls whether cells divide or not is something called density-dependent inhibition, or it's also called contact inhibition. It has two names. And basically the idea here, if I have a bunch of cells in a dish, so they're anchored to something, so they're going to divide, in this case, they'll keep dividing until they've formed a single layer across the bottom of the dish. However, once they're touching cells on all sides, so once they have no more room to grow, they're not going to divide anymore. So we say that they're density dependent, right? Once they've reached a certain density, once there's no more room to divide, they stop dividing. We also call this contact inhibition because once cells come in contact with something, they won't divide either. So cells keep dividing until they touch one another. Now you could also remove some of these cells and the cells would start dividing again to fill this hole. So this is good because in general you don't want cells to divide and clump up on top of one another, because that is actually what a tumor is when you have cells who are ignoring density dependent inhibition. So we can do a test to make sure that cells are properly following density dependent inhibition. That's called the scratch test. Basically the idea here, if you put some cells in a petri dish, they'll grow until they they'll divide until they form a single layer. And then if you scratch a piece out of the middle, they should divide to fill that hole again. So that scratch test, what we're looking at here, is a test for density-dependent inhibition or contact inhibition. Okay. So those were all things that allow cells to divide or not to divide that are not protein-based. But for the most part, the main signals that cells follow to know whether or not to divide are whether or not they are in the presence of signaling proteins. So these signaling proteins we call growth factors, and they're basically proteins that are released by other cells that stimulate cell division. So for example, if I was in the lab and I had two cultures of cells, so two different groups of cells, and I put some in the presence of no growth factors and some with growth factors, the ones that have these proteins that tell you to divide around, kind of logically, they're the ones that divide. So these proteins, these signaling proteins are really important to signal cell division. Okay. So this process is actually really, really complicated. So you can see here, we have the cell cycle. So we have G1 where the cell grows, we have S where DNA is replicated, G2 where the cell grows a little bit more, and then we have mitosis where the cell actually divides. And on here, I've put just some of the proteins that are responsible for controlling this process. So look I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I have 13 proteins here. And this is still an oversimplified version. So I just wanted you to appreciate how complex this process is. There are lots of proteins that control whether or not the cell can go from one step of the cell cycle to the next. And as a fun fact, Dr. Miller, one of the biology teachers here at Westwood, did her PhD research on this protein right here. Okay, so what these growth factors or these signaling proteins do, for the most part, they act as checkpoints. to determine if cell division is allowed to go to the next stage. So for example, I've marked 3 key checkpoints here on this cell cycle. We have the G1 checkpoint we're going to talk about, the G2 checkpoint, and the metaphase checkpoint. And at all these points, say the cell is going along, so the cell's gone through G1, and it gets to the G1 checkpoint. And these proteins, they check to make sure the cell is ready to move on. If it passes the checkpoint, it will continue on with the next part of the cell cycle. But if it fails the checkpoint, what the cell does, the cell no longer is in the cell cycle, and it goes into a non-dividing state. It's not allowed to divide. And we call that state G0. You can see G0 right there in my picture. So let's walk through these 3 checkpoints and see what we're actually looking for, what the protein complexes are checking for at each stage. Okay. So at the G1 checkpoint, and by the way, this checkpoint is controlled by the E2F and the RB proteins. Do not memorize those names. That's not necessary. Okay? And what they're checking for, they're checking to make sure that growth factors are present. Because remember, you were only supposed to divide if growth factors are present. So if they're not present, the cell should not be dividing. At this checkpoint, we're checking to make sure that the cell has adequate space to divide. Remember, we have to follow density-dependent inhibition. So we only want to move on if we have enough space. And then it's also checking to see if the DNA is intact and has no major mutations. And this makes sense because remember, after G1, if I pass the G1 checkpoint, I'm going to move into S. And S phase is where my DNA is copied. So if I have broken or mutated DNA, I don't want to copy that because then I'll have 2 sets of broken and mutated DNA. So this checkpoint is really important because we're making sure the DNA is okay before we copy it, because we don't want to make lots of mutated or broken copies of DNA in our cells. So you can see, here's a schematic of this. You can see here's the G1 checkpoint, and at this point we're checking, is there enough space to divide? Are there growth factors? Is my DNA mutated? And if we get all the correct answers, then we can move on. But if not, the cell is forced to stop dividing, and it either does not divide, or if it has mutated DNA, that cell might be destroyed by the body to get rid of it. Okay. So the next checkpoint in G2, so remember G2, this happens after S. Okay, so remember in S my DNA was copied, so in G2 I'm checking was the DNA copied correctly. Because again, if it wasn't copied correctly, I don't want to move on. So in G2 we're checking to make sure that the DNA was copied correctly. And then finally, the last checkpoint is the metaphase checkpoint. Sometimes students think that M stands for mitosis, but this is actually a checkpoint in metaphase, which is part of mitosis. And what we're checking for is that the kinetochores are correctly attached to the spindle fibers. So let's do a little review. Remember, when I have a cell in metaphase, all the chromosomes are lined up down the middle. Now remember, the spot where my chromosome is going to attach to the spindle fiber, that's called the kinetochore. So if I did a little blow-up picture of one of my chromosomes here, this spot right here, that's the kinetochore. That's where the spindle fiber is going to attach. So here, I'm checking to make sure my spindle fiber is attached correctly, okay? Because now, imagine if they weren't attached correctly. So I'm going to draw one that's not attached here, okay? So the reason this is important, because, this guy, and this, let me erase. Okay. So this one up here, this one right here, and this one down here, these are going to, those are attached correctly. So those are going to split my X's in half when the cell divides. But this one right here, because that's not attached, this entire X will go to this side of that new cell, which means one cell is going to end up with extra DNA, and the cell that divides on this side is going to end up missing this DNA because it's not attached correctly. So this checkpoint's important because we make sure the spindle fibers are attached correctly so that my chromosomes get split correctly. Okay, so we put this all together. In G1, we're making sure that my DNA has no mutations so I can divide. In G2, we're checking to make sure that the DNA replicated properly in S. And then in metaphase, we check to make sure all the chromosomes are lined up properly with their spindles attached to the kinetochores. So that's it for video 3, and I'll see you in video 4, where we'll talk all about cancer.